All right, AMP1 students, in this video we are going to talk about the function of the nucleus. So the function of the nucleus is to store genetic information, which it does that because that's the location where the DNA, so hopefully you can see this is DNA, a strand of it here. That's where our genetic information, our genes, that each of these little segments of our DNA, there's multiple genes along the segments of our DNA. That is the storage of our genetic information. And then what that genetic information will allow is for the production of our proteins in our body. And so the DNA has the information in order to make those various proteins in the body. So it's what is controlling the protein synthesis, which just means synthesis, just a word that means production. So it's what's controlling that. So there are two main processes that happen to allow for the DNA to be read. And then a protein to be formed. So it's a few steps in order to get from the DNA to our protein product. So we break it down into two main processes, and these processes happen in different areas of the cell. So like transcription. So hopefully you can tell that this here is the inside of the cell. So this whole screen, we're looking at the inside of the cell, but we're kind of zooming in so we can just see this cytoplasm inside of the cell here. And then this kind of lighter blue color is, you can kind of see the inside of the nucleus. And so this is the nuclear membrane. So there is like a membrane that surrounds the nucleus. And so it holds the DNA, keeps the DNA inside the nucleus. So it is able to protect that DNA that's in our nucleus. But there is little small holes in it, which needs to be, but they're definitely too small for the DNA to get out. So, all right, so that is what we're looking at picture-wise here. So the transcription process, it happens first. It happens inside the nucleus. And then the second process that happens, it happens in the cytoplasm or outside the nucleus. And it actually happens at ribosomes. So that was our function of ribosomes, was that ribosomes are the site of protein synthesis, and that's true. And so this is showing a ribosome. There's actually two parts to a ribosome. So you get this part up here and this part down here. But that's where translation is going to happen, is at the ribosomes that are in the cytoplasm. Okay, so in the learning objectives, we have the steps. I have the steps broken down for these processes. And we also have a video that helps you kind of visualize this process to help you remember it and understand it a little bit better. And then this picture hopefully will help as well. There are some pictures in the textbook too, but they sort of zoom in and give a little more detail than you need to know. And it's a little bit hard, I think, for students to tell where it's happening at, and I think that's important to know where these processes are happening at. So I like this picture a little bit better than what's in the textbook, so that's why I'm using it. Okay, so for transcription, what happens is there is an enzyme that will be necessary to, they call it unwinding of the DNA. So you can see here where the hydrogen bonds are formed, and that's keeping the DNA in that double helix. But you can see in this segment here, so this would be like this right here would be a gene, which is one segment of a DNA. You can see here where this gene is, the hydrogen bonds have been broken, and so they call this unwinding of the DNA molecule. And it took an enzyme to do that, so an enzyme was involved in breaking those hydrogen bonds there, so we could expose these bases here on this side of the strand of DNA. All right, so after that happened, that's kind of the first step. Then we had another enzyme, which they don't show the enzyme on here, unfortunately. But so just kind of imagine. There is an enzyme called RNA polymerase, so it's going to end in ASE because it's an enzyme. And what it does is it will attach to, and it has it will attach to the beginning of this gene. They call it like the starter segment of the gene. And so this RNA polymerase knows how to recognize those starter positions. So the RNA polymerase enzyme will attach to the starter part, and that just kind of tells, kind of directs, you know, which bases we're going to read along here. And then it'll also recognize the stop segment, so when it needs to stop too. So that RNA polymerase is going to bind, and what it will do will direct the placement of these little nucleotides. So this is trying to represent the sugar, and in this case it's RNA that we're making. So there's like a little sugar, ribose sugar, and the phosphate, and then the base that's there. So there's a little we call it ribonucleotide, since it's a nucleotide that's going to form RNA. So you can see that these have already formed. So, and remember, the bases are complementary. So if there's the guanine, then a little ribonucleotide that has a cysteine will bind, form a hydrogen bond there. And then cysteine will, a little ribonucleotide that has a guanine will bind there, and then so on. And then you can see that this was one of the ribonucleotides getting ready to bond there, and also bond, they also form bonds between the phosphates too and the sugar. And so once we have, remember the RNA polymerase enzyme that they're not showing is directing this from the start to the finish. And so once we get to the, that RNA polymerase gets to the end of that segment of DNA or that gene and knows that it needs to stop, then it will stop, and what will happen is the messenger RNA, this one strand that was formed, it will detach. Those little hydrogen bonds will be broken, and it'll detach. And so then this little messenger RNA will, and this is representing it even though it's not really showing the bases. I guess it's just writing them on there. It will detach, and then it's small enough because it's just single-stranded and it's just a short segment. It's able to get through the nuclear pore, the little pores in the holes in that nuclear membrane, and then leave the nucleus and get out here in the cytoplasm and go find a ribosome. And what it's carrying with it is the instructions to make a certain protein in the body. So it's going to be this protein, but we're not making it a specific protein, just a regular protein. So that is the steps in transcription. So basically what you're doing is starting with a DNA molecule and forming a messenger RNA molecule from that process. Okay, so now I'm going to go to our learning objectives. Okay, so the learning objectives for transcription are 10 and 11. So 10 just goes over kind of what's going on in transcription, kind of the overview. So transcription is that process where you get the formation of a messenger RNA. I don't know if I mentioned that. They call that little RNA the messenger RNA because it's going to take that information from the DNA and take it out to the ribosome. So it's kind of serving as a little messenger with the instructions. And it's going to take that message to the ribosome so we can make a protein. And this whole process occurs in the nucleus. And so we start with the DNA molecule, and our product in this case is RNA. And these are the required structures. You need DNA, you need the RNA polymerase enzyme, and the ribonucleotide bases, and then the messenger RNA. So if you got those things, then transcription can happen. And so these are the steps that I just went over with the picture for that process. All right, but we're not done yet because we don't have a protein yet, and that's what our end goal is to get a protein product. So then we're going to talk about translation. Now, for the process of translation. So now that we have our messenger RNA that was formed through that process of transcription, it will head out to the cytoplasm, and a ribosome will bind to that messenger RNA. So this little messenger RNA is now attached or bound to this ribosome. And you can see the little base pairs again. All right. So if you will notice, remember that the building blocks of a protein are amino acids. So we need to get amino acids bonded together so that we can make a protein. So one of the molecules that we haven't talked about yet, it's called transfer RNA. So it is also an RNA, short little RNA molecule. And what transfer RNA has associated with it is it has some base pairs in a certain order. So this one's GCA. And because it has that particular base pair sequence, it is carrying a particular amino acid. So this is a blue amino acid, so we'll say it's phenylalanine. But then you can see this transfer RNA, so this is a transfer RNA as well. It has the base pair CAC. So it is carrying this little turquoise amino acid, and so that could be like lysine. That could be the amino acid lysine. So, and then this would be a different amino acid because it's a different sequence of these three bases here for transfer RNA. Okay, so when that ribosome attaches to that messenger RNA, then what will happen is the ribosome will hold that messenger RNA in place, and these little base pairs and it goes by threes because the transfer RNA has three base pairs, so they call those codons. So that's a codon. This particular one is going to be complementary to this particular transfer RNA, and it'll have that particular amino acid. So because the messenger RNA has this particular sequence of base pairs, so it looks like GUG, then a certain amino acid will bind there. So each of these transfer RNAs always carry the same amino acid depending on their base pair sequence. So this is always lysine, it has a base pair of CAC, and this is always phenylalanine because it has a base pair of GCA. So they will bind, and the reason they're binding through hydrogen bonds is just to get that amino acid in place. And so then once the amino acid is in place, so they got it in place here, and once that amino acid forms a bond with the adjacent amino acid, then you don't need that transfer RNA anymore, and it will release. So it'll release its amino acid, and it'll also release from the messenger RNA. And then it'll go off to find another amino acid that is specific for its base pair. So it has a certain shape, so it can only bind to a certain amino acid and hold it. All right, so now that we have and this will continue on, so now that we start having these amino acids being placed in the proper position thanks to the transfer RNA binding to the little codons of the messenger RNA, then our little protein link of our little polypeptide, they call it, growing protein chain, it will just get longer and longer and longer and longer. And then once the ribosome actually notices when the stop codon is reached, and then it will stop, and then it'll release that messenger RNA, and then it'll also release that all those amino acids that are linked together, so that growing protein chain or polypeptide chain. And then remember, our proteins will not kind of stay in that linear form. They'll fold along themselves to become a secondary structure and then a tertiary structure, and then they'll form with other proteins, polypeptides, and then they can become like a quaternary protein structure that has a specific shape that can perform a specific function. So that's the steps in translation. So it happens in the cytoplasm. Your beginning product is the messenger RNA, and then your end product is the protein. And then the players in it are the transfer RNA molecules that are, along with the amino acids and the messenger RNA. They're allowing the formation of that long polypeptide or growing peptide protein chain to form so that we can get a protein.